Hi, I'm Beatrice Setnik, Chief Scientific Officer at Altasciences. Thank you for your interest in our poster titled Practical Considerations for the Evaluation of Physical Dependence and Drug Withdrawal for Novel CNS Active Drugs in Clinical Trials. This poster is a collaborative effort with other members of ISCTM, which is the International Society for CNS Clinical Trials and Methodology, who have a working group established to discuss and present ideas on evolving clinical trial methods. And what we are focusing on in this particular poster is the issue of pragmatic collection of data for novel CNS active drugs to evaluate whether or not they have physical dependence and associated withdrawal symptoms upon abrupt study drug discontinuation. These are requirements by the FDA, and they are addressed in the 2017 FDA guidance on abuse assessment of drugs. These are specific for drugs that are chronically administered, and oftentimes the agency will require sponsors evaluate any symptoms of drug withdrawal that can occur after a study drug has been abruptly discontinued in a clinical trial. So typically these are evaluated at the end of phase 2 or phase 3 type studies. However, we have an understanding that the requirement for assessment of abuse and physical dependence requires very frequent administration of measures and assessments because the withdrawal symptoms can occur a few hours after drug discontinuation, dependent on the half-life of the drug. In order to capture these pragmatically, we need to think about some modifications to the study designs themselves in order to be able to accommodate and keep patients engaged in collecting these measures, even though they are technically have discontinued the study drug and may not always be motivated to continue collecting study assessments. So in these cases, we are looking at a variety of different methods. And one of the key issues that we find in these trials that are limiting are the fact that many of the assessments that assess drug withdrawal symptoms, as you see in Table 1, are actually clinician rated. So Table 1 gives you an example of a schedule of time and events that could be imposed in a phase 2 or 3 clinical trial, and it can show you the need for very frequent assessments. However, much of the evolving methods that we're proposing here in this poster may address some of those limitations that are operationally more challenging to administer. So for example, the clinician-rated scales that currently exist and are valid are all mostly clinician-rated, as you can see in Table 2. The issues that we have are that once they are clinician rated, these are really limited to in-clinic visits or telehealth visits where a clinician can assess a patient. And as you can probably recognize, these are very difficult to administer on a daily basis when they're requiring clinician oversight. And so therefore, a pragmatic solution to this problem is to develop these types of scales into subject-rated scales where patients can do these on a daily basis with some type of media or digital electronic diary. And in fact, this is a tool that's under development within our groups to have a more pragmatic, self-administered clinician subject-rated scale that will allow for the monitoring of drug withdrawal symptoms. In addition, there are other approaches that we can look at with evolving technologies, and certainly medical devices and new wearable devices can really help us obtain important physiological data, safety data in these types of trials without having to rely on in-clinic visits. And the evolution and the ongoing improvements in these technologies allow the integration of these into clinical trials and offer another solution that will allow us to monitor these patients from a safety perspective frequently after they discontinue a study drug. The same is also the case for pharmacokinetic sampling. Microsampling and advancements in microsampling have enabled us to allow for much lower blood volume collections that can be collected remotely to look at the plasma drug concentrations on a more frequent basis in these types of trials. So a number of areas that have been evolving and new scale development will really help us implement more frequent assessments to really characterize any potential risks that novel CNS active drugs may have before they are marketed. And so in conclusion, if you take a look through the poster, you'll notice that we do address and provide some examples of the emerging technologies that can be implemented and the tools that are currently under development and construction for this. If you have any questions, please feel free to contact us and be part of our ISCTM working group and get involved in the discussion on these novel approaches for clinical trial methods. Thank you.